Community Health Research Exchange. We're going to be recording today's talk, so if that's okay with all of you, please click the Got It button as it pops up. Today, we are really excited that we have three of our outstanding K scholars who are going to be presenting their research. These wonderful scholars are supported through the Institute for Clinical Translational Science Training Award. And so for those of you who are new to UCI, this is a large grant mechanism that supports clinical and translational science here at the university. A big welcome to all of those who joined from the community. We're excited to have you. Our K scholars today developed these talks to be really accessible. So we're excited to hear your thoughts on the research as they present it. We will have a short 3-question survey that will pop up after each of our scholars' presentations. It will come in the chat. So if you could complete those as you go, that would be wonderful. This is really important feedback for our scholars so that they can continue to develop their public speaking skills. If you have any in-depth feedback that you'd like to share, please feel free to email Robin Zender, who is on many of the communications, and her email is rzender@uci.edu, and we can put that into the chat. The mission of the CHRE generally is to bring together community and academic researchers to explore all aspects of community-engaged research. And we're really excited because this is the first time that we're asking our scholars to present as part of the Community Health Research Exchange. Just one last thing before we get started, I want to acknowledge some thank yous. The first thank you is to our 3 scholars, and we'll have a chance to thank them again as they go through their presentations. But I also want to thank the team that helps support developing the presentations and supporting our scholars today. So a big thank you to Vince Cayoza, Marguerite Klum, Isabella Nardo, Margaret Schneider, and Robin Zender for all of the support in helping us get to this exciting day today. Robin Zender is the co-lead for she's the staff lead for the community engagement part of the ICTS. So if you have any questions about community engagement, you can reach out to Robin. And Pam Pimental is our community lead. And if you have for the community engagement core, so community folks or academic folks, if you'd like to reach out to someone with expertise on community academic partnerships, please reach out to Pam Pimental. Okay, without further ado, our first talk today will be from Dr. Zahedi, and she will be speaking about driving towards treatments for spinal cord injury by boosting cellular engines. And Dr. Zahedi is a assistant professor at the Sue and Bill Gross Stem Cell Research Center. So welcome.
Thank you so much for that kind introduction, and for Marguerite and Vince and all the colleagues for this wonderful opportunity to put this together. And I'm going to share my screen. Hopefully you guys can see my screen well. And I'll go ahead and get started. Yeah, so thank you for that introduction. As you mentioned, I'm a KL2 ICT scholar here working at the Stem Cell Research Center. And my talk is entitled Driving Towards Treatments for Spinal Cord Injury by Boosting Cellular Engines. Here you guys will see sort of an image of an injured spinal cord, where on the left side, actually the spinal cord has been injured and there's some loss of cells and tissue and scarring as well. But before that, I will sort of jump into some brief background. Here's an outline of my talk. I will begin with sort of my own personal background, giving you guys a little bit of sort of a view into that. And then I'll jump into introducing the mitochondria and how, when it gets damaged in injury and disease, that could be detrimental. And then we'll jump into talking about therapies for spinal cord injury. I will introduce neural stem cells, which are very promising candidates. And then also the option to combine mitochondrial drugs to enhance stem cell therapies. But some of you might be wondering, what is the mitochondria? You may have heard that they are the so-called powerhouse of this, the cell. But essentially, what that means is similar to how a car engine can use the fuel and then generate energy for the car to move. The mitochondria uses oxygen and nutrients and generates energy in the form of ATP for the cell to do a variety of functions. And that will be sort of the focus of my research, and I will talk a little bit more about that as well. Here's a little bit of my personal background. I actually have a younger brother who sustained spinal cord injury at the age of 2. And that involved us, the whole family, moving to the US at the age of 10 and seeking medical attention. So I've spent a lot of my time in hospitals. And so that sort of inspired me to join academia and become a bioengineer in undergrad, and got my master's and PhD at UC Riverside. Of course, there's a lot of innovations for spinal cord injury in the form of robotics or medical devices, but I quickly became interested in neuroscience and was really in search of a cure. So this led me to sort of study the mechanism of spinal cord injury a little bit better, and sort of join UCI, where I'm now studying stem cell therapies as a potential cure. So what are the mitochondria? Let's talk a little bit more about them. Here in the video to the left, you see a healthy neural stem cell, which has its mitochondria labeled. These are these small, sort of round organelles that are moving, and they're highly dynamic, and they change their shape. But as a cell gets damaged, whether in injury or whether it's been treated with some stressor or toxicant, the morphology of the mitochondria can change. And here you guys can see some of these more swollen mitochondria, which is sort of not a good indication. And it suggests that these mitochondria have been damaged and can cause the cell to essentially also become damaged and lose some of its key functions. And we alluded to the main function of course, generating ATP, but mitochondria are involved in a variety of other types of function, one key point being cell death, and a variety of other functions that I'll be talking to you guys about. And I really wanted to highlight too that mitochondria are really important. They're involved in a variety of different disorders, ranging from neurodegenerative to aging, all the way to cardiovascular disease or diabetes and even cancer. So they are quite important in almost every single one of your cells. And with that, I want to sort of now focus on my particular disease of interest, spinal cord injury. And just a few facts about spinal cord injury: roughly 250,000 new cases every year that we get traumatic spinal cord injuries. And roughly 50% of these are due to road accidents, followed closely by 24% due to falls. So it's a very debilitating condition, life-altering condition. And what does it look like, spinal cord injury in the actual tissue? Well, for us, if you look at the image in the middle, you see a healthy spinal cord. You see the white matter and the gray matter, and very nice architecture, everything intact. The spinal cord to the right has had injury done. You guys can kind of see the site of the injury where there is definitely loss of cells. But what also happens essentially is that the blood vessels can also get ruptured, and this results in a lack of those valuable oxygen and nutrients to the neighboring tissue. And eventually, over the course of time, a secondary injury sort of forms, and more cells can get damaged and essentially die here. So let's now talk about some of what are the potential therapies for spinal cord injury. I mentioned neural stem cells are a very promising candidate. Here's another image of a neural stem cell. Here, those mitochondria that I showed you guys previously that were moving around have been sort of segmented, each as an individual color, so we can distinguish them easily from each other and kind of see that they have different shapes and sort of different distributions and so on. So we did mention that, of course, spinal cord injury causes loss and damage to the cells. We have cells that are dying. And this is exactly why neural stem cells are so important, because they can generate new cells of the central nervous system. They can actually, here's an image of neural stem cells, which have formed 3 cells. In green, you will see neurons. In red, you will see astrocytes, which are the scar-forming cells. And in white, actually, you see oligodendrocytes, which are the myelinating cells that myelinate nerve bundles and allow for electricity to be conducted. So as I mentioned, neural stem cells are quite powerful. They can generate new cells. They can secrete factors that are protective for that environment. And so in this particular study, we asked, what if different stem cell lines vary in their mitochondrial behaviors, their functions, their quality, and that in turn can affect their repair potential? Meaning, what if not every stem cell, behaves the same? And I'll be talking to you guys about this option, the therapeutic solution of what if we can combine stem cells with mitochondrial drugs to then be able to allow them to have a healthy, resilient sort of mitochondria as they are placed in this sort of harsh spinal cord injury environment. But before that, I wanted to kind of jump back and tell you guys about these different human neural stem cell lines that were established and developed in the lab by a very, very talented researcher, Katya Pilty, shown here. She actually developed these lines, these UCI lines, and characterized them to see how effective they were in spinal cord injury. And the way she did that is she would transplant mice that had spinal cord injury with these different cells and then test them for different locomotor function to see how well do they walk after the spinal cord injury and are they able to recover. And based on this scale, here we identified some cells. For example, UCI 161 was our most effective line, shown here in green. And moving forward, we'll be referring to this as our good line. And we have our UCI 152 lines, which was shown to be not so effective. And so we will call this our so-called bad line or non-effective line. So that's exactly sort of where I joined the lab. And going back to that hypothesis, the question that, indeed, is it the different mitochondria sort of functions of these cells that make them different? And to kind of better study this, I came up with the terminology of mitochondria fitness traits. And here's a list of 5 that are listed here. And essentially, these are different functions or parameters of the mitochondria, as we talked about. It's not just one or just making energy. And so in the figure to the left, I'm showing you guys genes for only one of these traits called redox balance. And you might ask, well, what is redox balance? Well, many of you are probably familiar with the term that if you drink a glass of wine a night, you will, never be sick and you keep the doctor away. And that's essentially due to the presence of resveratrol. It's a molecule in wine that is an antioxidant, essentially is a protective agent against stress. And here we have different genes related to oxidation and antioxidants. And what I want you guys to really appreciate is that these 2 cell lines, 152 and the 161, are good and are bad lines, have varying amounts, essentially, or levels of these genes, which can make them become more resilient or less resilient to stress. And we'll be talking about one in particular, NRF2, which I've highlighted here next. NRF2, similar to resveratrol, is actually an antioxidant molecule. It helps to combat oxidation. And when we looked at our cells and we stained for NRF2, we see that in our good line, our 161 line, NRF2 levels are higher or brighter, as you can see by this fluorescent staining, compared to our not good cell line, which are lower. So this perhaps suggests again, that our good cell line are able to be more resilient to oxidative stress, which is indeed what they will encounter in that spinal cord injury environment. So now let's jump into this option of mitochondria drugs. If there are certain lines that are less resilient, can we perhaps enhance them, boost them by combining the use of mitochondria drugs as a therapeutic? So to do this, we sort of dug into the literature. We actually combed through clinical trial data. We looked at FDA-approved drugs. We wanted to really work with drugs that have already been shown to be safe and have some level of efficacy in various diseases. And we identified them from our 5 different traits of mitochondria function. And so you see them all listed here. And again, through a series of tests, we were able to identify one very promising drug called bezafibrate. Essentially, this is a lipid-lowering drug, so a metabolic drug. and it's in the category of biogenesis. Biogenesis, it really means the creation of new mitochondria. So when we tested bezafibrate on our cells, we treated our cells for 2 days in the dish with bezafibrate, and we showed using this TMRM analysis that corresponding to higher levels of intensity that you notice in the bezafibrate treated cells, these cells have the capacity for greater levels of ATP production. We also showed that they had larger numbers of mitochondria, which makes sense because this is indeed a biogenesis drug, and that's the creation of new mitochondria. So with these very promising results in the dish, we decided to sort of further test this hypothesis in animal models. And to do this, we again took bezafibrate pre-treated cells and we transplanted them into spinal cord injury mice. And we used another locomotion test here, referred to as the Basel Mouse Scale, BMS task, where we're looking at different aspects of a mice walking patterns, coordination, and so on. And we rate the mice's score based on 0 to 9 here. And here, I hope you guys can appreciate that previously our 152 cells, which are bad cells that were non-effective, were actually able to become effective and allow these mice to gain greater levels of locomotor recovery. Interestingly, our good cells actually had a little bit of harm and they actually lost some of their effectiveness. So this was a very exciting yet sort of interesting finding for us. And currently we're in the process of further investigating this by looking at the tissue in the mechanism in a further way. So I wanted to sort of wrap up some conclusions here. I hope I have convinced you that the mitochondrial functions of different neural stem cell lines can vary between especially effective and non-effective lines. We talked about the effective UCI 161 lines, for example, having higher levels of NRF2, which was a protective molecule. We also talked about screening and identifying these clinically relevant drugs. And we showed that when we treated our lines with bezafibrate, we were able to increase their energy capacity as well as increase the number of mitochondria that they had. And when we took the bezafibrate pretreatment cells into mice models and we transplanted them into spinal cord injury mice, we actually showed that we were able to improve the recovery of locomotion. And currently we're in the middle of exploring this mechanism. And we think it might have something to do perhaps with differentiation capacity of these cells being changed. So that will be exciting stuff to sort of look into further. And I just wanted to end with some just general broad implications that, aging in general is very much a mitochondria-related disease and affects the whole body. So perhaps mitochondria drugs and as well as stem cell therapies can be very promising therapeutics for the greater sort of community as well. And with that, I wanted to definitely thank all the people that have contributed to this, my collaborators, my PI, Dr. Eileen Anderson, all of the funding agencies, especially the ICTS community for this opportunity and this funding to allow me to do this research, without which none of this would be possible. And with that, I can definitely take some questions if they are available.
Thank you so much, Dr. Zahedi. That was really lovely. Thank you. Do we have questions?
Yes, I have a question. First of all, how is your brother?
Oh, yes. So my brother is doing well. He definitely still has spinal cord injury and a variety of other issues, actually, a lot of muscle loss as well. And but he manages to be very independent. He drives, he lives in Chicago with his girlfriend and dog. Works at a disability sort of nonprofit where they help the people of Chicago sort of make the community more accessible, and is leading a very fulfilling life. And
that's great.
Yeah,
good to hear. Some more questions unless anyone else. Okay, so I just want to make sure I understood. So the drug bezafibrate is actually creating new mitochondria? It's actually allowing the body to create new ones.
That's correct. That's correct. So it is generating new mitochondria, which we thought would be a better approach than sort of there's other drugs that can force the mitochondria that you already have to just produce more energy. It's kind of like making workers in a factory work double shifts, for example. You would get double the output. Bezafibrate is the equivalent of building a second factory. So you basically get double the output, but not overload of the mitochondria. So it's very safe and sort of to the mitochondria as well. Yeah.
Okay. And my last question, or maybe last question, is bezafibrate currently being used as a treatment modality for spinal cord injuries, or are these not even available to the public yet?
Yeah, so I think I may have not mentioned in detail, bezafibrate is in clinical trials in the United States and is FDA approved in Europe and across other areas for predominantly lipid lowering sort of applications, basically for diabetes. It is sort of administered and so on. But it is gaining a lot of interest because it has a lot of properties. So people are very much interested in applying it for spine. But to date, there is no trial yet, no clinical trial yet with bezafibrate in use with spinal cord injury. But it is definitely gaining a lot of traction, lots of clinical trials being done in immune-related conditions too. And it's interesting because spinal cord injuries is also a type of injury where your immune cells also get activated. So we think they might have some indirect roles there. Yeah.
Yeah. Thank you. That was really an interesting talk. And I'm thinking of in the future, you're by the bedside of a 2-year-old and their family, and you're trying to explain all of this. And you know, you would, I think, if you were giving this to because I'm not sure, I'm a little bit of a in the healthcare field as a retired nurse, but you would have to really start with the basics, right? Of exactly what is the spinal cord and what's its function and what's all the parts. Yeah. But and the last, I'm sorry, I have another question. What are the side effects though? So the little mouse was able to walk better, but did its tail fall off? Or I mean, are there other things that happen as a result of using this medication?
Drug, yes. So actually the only side effect, which is sort of an intended side effect for its original use, is bezafibrate does have a little bit of a weight loss side effect. So
can I sign up for this drug now?
yeah, definitely. That's actually my whole lab was like, we could, put this on the side, but yeah, it does have a little bit of weight loss. We have to be very careful with spinal cord injury mice because of course they're injured. So we don't want to also cause them any unnecessary sort of weight loss. But yeah, so that is the only possible sort of like side effect. It is very, very safe. Part in due, as I explained, why is sort of it doesn't overload your current system. And it
kind of makes sense, more steady, right? With the whole mitochondria increase, which is the engine that you would have, a better metabolism.
Two engines, basically, yes.
Yeah, and I can see how that mitochondria would also improve the other diseases that it's currently being treated for. Thank you so much. I really enjoyed your talk.
Thank you so much, Pamela.
Thank you. Do we have any other questions? That was excellent. Thank you very much. Please, if you have a moment and didn't see it pop up in the chat, to go ahead and fill out the survey. Please click on the link. It's in the chat. And there are 3 questions. Thank you. Give you a moment to do that.
And do you mind if I share my screen?
Please do.
Okay.
Okay, our next presentation will be from Dr. Hicks, an assistant professor in the Department of Physiology and Biophysics. And the title of his presentation today is Stem Cells for Muscular Repair. Welcome.
Thank you very much. Thank you, Vince, Marguerite, and Robin, for giving me the opportunity to speak today. And thank you all for attending. I'm an assistant professor here in Physiology and Biophysics and the School of Medicine. And my lab started here in 2020. Before that, I did my postdoc at UCLA, and I'll be giving an overview of some of my research that I've been pursuing in my independent position, as well as where I started in muscle repair. and I hope to tell you more about the general overview of muscle and stem cells and where we are as a scientific community today. So this is a picture taken from my website showing the different areas that my lab focuses on. We focus on pluripotent stem cells, which can form any cell type in the body, the stem cell niche, which is the home for where stem cells reside, and we apply these to different neuromuscular diseases with innovative new stem cell technologies. And so when I was a kid, I played a lot of team sports, and I would fall down, I would dive and jump, and I would take for granted how well our muscles repair. We have an injury and we may not even think about how much our muscles repair, but skeletal muscle is central to everything that we do. It's important for our ability to move, our ability to even sit down. We would fall over without having posture holding us upright in our chairs currently. For chewing and swallowing, breathing, that our diaphragm controls our ability to breathe. Skeletal muscle regulates our body temperature and our metabolism. And so skeletal muscle really has broad functioning to life. And without skeletal muscle, we would be disabled, we age, and we would die. So skeletal muscle can control limb defects during development, such as dysplasias and skeletal dysplasias. And as we age, we undergo age-related muscle loss called sarcopenia. And there's different genetic diseases that are affected by skeletal muscle. This is famously Stephen Hawkins. This is actually a genetic disease of the motor neurons, ALS, but the muscle is innervated by the motor neuron, so it also just undergoes dystrophy when your neurons are not functioning correctly. And so, as I mentioned, skeletal muscle is one of the most regenerative tissues in our body. It's quite fascinating just how regenerative the skeletal muscle is. And so you can see over here a healthy skeletal muscle. This is a cross-sectional area of a skeletal muscle. If I were to chop it the same way you would a bread loaf, it's a transverse section, and you can see the muscle fibers here. The muscle can be almost completely destroyed. And it can regenerate over a period of about 4 weeks to become almost exactly healed the way it was. And so you can just appreciate how damaged this muscle can actually be. And the reason why the skeletal muscle repairs is because of the endogenous muscle stem cell that resides nearby the muscle fibers. So muscle fibers are ginormous muscles. They're ginormous cells. Think about your bicep. They run from your shoulder all the way to your elbow. There's one cell that is that large. And the muscle's primary job is to contract. And so to contract, it must have multiple nuclei, like 1,000 nuclei. And you think about cell biology, each cell has one nucleus, but the skeletal muscle has thousands. And so because of that, it cannot undergo a cell division, and it requires the muscle stem cells, which are nearby the skeletal muscle, in order to support it and to fuse and to make new nuclei to support the muscle fiber. And so the muscle stem cells, upon injury, these cells express a transcription factor called PAX7, and this is a really important transcription factor that gives the muscle stem cells its ability to reside in a non-proliferative state, and it can live in your body for as long as you live, till you're 100 years old. These stem cells are there, and upon injury, the stem cells will activate and proliferate. They'll turn on different myogenic genes, and they'll differentiate to repair the skeletal muscle. And at the end of regeneration, a subset of those muscle stem cells will return to their home called the niche, and they'll reside in this non-proliferative state, and they're waiting there for another injury that you may experience months or years later. And so this is a process called self-renewal that the muscle stem cells are able to accomplish to repair muscle. However, there are some instances where the injury or the disease is just too severe, and the muscle stem cells try their best. They will repair it as much as they possibly can, but eventually they become exhausted or they're dysfunctional. And when this occurs, you have chronic degeneration of the skeletal muscle. And so this really comes into play where research is needed to either reverse the exhaustion of the muscle stem cell, or provide the muscle with donor stem cells that can then replace the exhausted or dysfunctional stem cells. So I like to put this slide on there because maybe everyone doesn't think so much about what actually is a stem cell. We think about the word stem cell, it's a fun buzzword, but what actually does a stem cell do? So the quintessential definition of a stem cell is something that can self-renew. And self-renew is when that stem cell can divide to make a daughter cell that is the exact replicant of the parent cell. And a stem cell must make an important decision. Do I divide to make an exact daughter cell of myself, or do I differentiate? And so there is different stem cell systems across our tissues that can make different tissues. For example, our blood and our skin have stem cells. And so there's a bifurcating event that tells a stem cell to differentiate or to self-renew. And this is actually a really hot topic in the stem cell field, is how does this actually controlled? And there are two primary types of stem cells. You can think about the pluripotent stem cell that occurs during fertilization, and these pluripotent stem cells can make every single cell type of the body, like we do when we're newborn babies. And these form the blastocysts. You could think of this like in an IVF that can go on to differentiate to, say, skeletal muscle or neurons. And we can actually take these type of pluripotent stem cells and generate them in a dish and grow them up indefinitely, and then try to control their method of differentiating them to these different tissues inside of a dish. And this is a really hot topic that our lab is studying for skeletal muscle. And then there's also adult stem cells, and all of us have adult stem cells. These adult stem cells have limited potential. So skin can only make more skin, and blood can only make more blood. But skin can't make liver. They can't differentiate into other lineages. They're lineage restricted. And many of these stem cell systems are rare and they're difficult to obtain from humans. However, there are some examples where adult stem cells are being used in clinical trials. And one example, this is work from Don Cohn's lab at UCLA. This is an example where stem cells have actually been used to cure immunocompromised human disease called bubble baby disease. You may have all seen the Jake Gyllenhaal movie where the bubble boy is inside this literal plastic bubble because his mom was afraid for him to get sick. But this is actually a real disease where your blood stem cells aren't functioning correctly and you have no immune system. So Don Cohn's lab was able to take these blood cells, give it the healthy genetic material to those cells outside of the body, and then infuse those back into a person and actually cure their immunocompromised disease. And this is a great example of how stem cell therapies have actually been used to cure people. Now, as a stem cell biologist, I also like to put this slide on there, and I think this is really important. There are over 2,700 unproven stem cell clinics across the United States. And when I tell my old high school friends that I'm a stem cell researcher and they say, oh, I'm getting stem cell injections for an unproven therapy, unproven disease, I lay this out as caution that the FDA does not regulate stem cells because they're not a drug. And so people can get these therapies, and probably what's going to happen is it's just going to cost them a lot of money for a very little effect. And so as a community, I just caution this as, please do your research on going for different types of stem cell therapies because their effectiveness, if you want to get the FDA approved stem cells that have undergone clinical trials like that we're doing here at UC Irvine. So in 2007, this person here, Shinya Yamanaka, discovered a really remarkable scientific discovery, and that is that you can make pluripotent stem cells from adult. And so he used what is now coined in the scientific community as the Yamanaka factors, a set of 4 genes that can reprogram an adult cell back into your embryonic state. And these are called induced pluripotent stem cells. And if you have an induced pluripotent stem cell, it can go on and make any cell type in the body, just like early fertilization. And we can use these for disease modeling because I could take your cells, reprogram them, and they still are exact copies of you. So they have all of your genetic memory. If you have a mutation or such, those cells will also have it now in a dish. And so he won the Nobel Prize for this in 2012. And we in our lab now use these induced pluripotent stem cells for studying all kinds of muscle diseases, and we can make skeletal muscle from them. So these are pictures of induced pluripotent stem cells from my lab. And they express this marker here, OCT4, which is an embryonic stem cell gene. And we can insert different genetic material into those induced pluripotent stem cells. So here we put a red fluorescent protein so we can actually look at the cells and watch them under a live microscope with fluorescent microscopes. One thing that we've done is we've cultured the induced pluripotent stem cells on different types of extracellular matrix from either healthy people or from people with muscle diseases, and watched how they migrate and grow. And so you can see on the wild type healthy matrix, these stem cells seem to integrate really well. Whereas on this dystrophic matrix, they seem to just grab a hold of the matrix and run around. They migrate, they don't integrate with the matrix very well. And there's definitely an issue with the matrix there. And so we're actually using induced pluripotent stem cells to study different diseases. And one of the diseases that we're studying in our lab is called Duchenne muscular dystrophy. And Duchenne muscular dystrophy is an X-linked disease that affects boys, and it's a progressive muscle disease, and there's no cure for this disease. And I like to think of Duchenne muscular dystrophy as a cable bridge. So the muscle's most important thing is that it needs to contract, and the contraction occurs in these proteins called the sarcomeres, where you have myosin and actin that cause contraction. And dystrophin holds the sarcomere to the outside of the cell. And on the outside of the cell, you have this complex called the dystroglycan complex. So dystrophin is this steel cable. And if you don't have that steel cable, when you contract, the muscle will rip in half. And when it rips in half, calcium ions flood into the cell and they kill it. Now, the muscle stem cells will do their best job to repair the myofibers. So that's why boys can live at all. But those muscle stem cells eventually exhaust, and then you run you run out of them, and these boys are currently dying. So what we've used is something called CRISPR-Cas9 in the induced pluripotent stem cells taken from boys with Duchenne, where we can use this gene editing material to remove the mutation from the dystrophin gene, which allows for the dystrophin gene to now be transcribed in what we call the corrected Duchenne muscular dystrophy, iPSCs. And it looks like this. So over here, what you're looking at is the exons, and these exons are part of your DNA that assemble to make a gene. And in dystrophin, you have 77 exons. It's a very, very large gene. But we can use CRISPR-Cas9. So how that works is you have an RNA that serves as a tag, and it can go anywhere inside the genome and bind to a certain area. And then the Cas9 is like a scissors, and that scissors comes in and chops where that tag is located. And so we can chop out a region of the dystrophin DNA, and then it will recombine to make a smaller dystrophin, but it's a functional dystrophin. And so there's actually a clinical disease called Becker's muscular dystrophy that's extremely mild. And some people never even realize that they have this Becker's muscular dystrophy. So we can change a Duchenne muscular dystrophy cell into a Becker's muscular dystrophy, which is very mild because it has a shorter but functional dystrophin gene. And so we use this CRISPR-Cas9 to generate that. So as I mentioned, Duchenne muscular dystrophy lacks a protein called dystrophin. And so these are cross-sectional areas. The dystrophin muscular dystrophy has no dystrophin, as shown here. The Becker's actually expresses it, and this is the normal human. So you can see that the Becker's muscular dystrophy is much more similar to the normal human patient as opposed to the Duchenne. And so this is our overall goal in my lab, where we have patient-derived cells that we can reprogram into induced pluripotent cells, use CRISPR editing to correct the Duchenne disease cells, and then differentiate these into skeletal muscle, and then study their ability in mice to either restore the dystrophin protein or replace the exhausted muscle stem cell pool, because the muscle stem cells in Duchenne are no longer functional. They've been exhausted over time. Another way that you can actually deliver the CRISPR-Cas9 is by packaging it into something like a virus. These viruses are called AAV, and they're in clinical trials for different areas like the eye, for macular degeneration. So you can package the DNA material into a virus and then directly deliver this into boys. And this is pursued by my postdoctoral lab in Myogene Bio that started a company to just deliver this straight up. But what we've really focused on in my lab is making these cells, these skeletal muscle stem cells for cell therapy. And so the process for differentiating a pluripotent stem cell into a skeletal muscle takes us about 6 weeks. And this is actually really challenging, because to make a pluripotent stem cell into a muscle, you have to actually follow the way that it occurs in human development. You have to know about how does human development work, and then use those developmental cues to coax the cell at every single step along the way. So it has to make the specific lineage, like the mesoderm, and then the somite, and then the myotome. And these are words that you might know if you're, a developmental biologist. And these are things we've had to really learn on how each one of these different human cues develop over time. And so what you can see is that we've accomplished this. And the Duchenne muscular dystrophy, these are cells in a dish. These are skeletal muscle cells in a dish expressing myosin heavy chain. So you can see that we can efficiently make these myosin heavy chain positive cells in a dish. And in the CRISPR corrected lines, we can restore the dystrophin that's missing. And we also show here in white that we can now generate the muscle stem cells inside of these dishes. What's really cool is we can isolate these muscle stem cells, which are shown here in white. We can isolate those, and we can use those for cell therapy. And we can then transplant those for what we do currently. We transplant into mice that have the human model of muscular dystrophy to see whether we can restore the dystrophin protein. And that's actually what this looks like here, where we differentiate our pluripotent stem cells into the skeletal muscle progenitors that express PAX7. We transplant these into a mice, and after 30 days, which is about the time that it takes for a muscle to regenerate, you can see here in red are all of the human nuclei that we've transplanted, and in green is the missing gene, dystrophin. And so we're able to restore dystrophin in mice models to very high levels. And so what I've shown you today is that stem cells for muscle repair can restore missing proteins in a dish. They can improve muscle function inside of a mouse. And this has a very wide range of applicability for different types of disease. We've studied this for Duchenne, but this could actually be applied to any muscle disease or injury or aging. There's many different ways that we're applying this. And so I've actually teamed up with a number of faculty and both basic science PhDs and MDs here at UC Irvine. And just this year, we formed a new center called the UCI Muscle Biology and Disease Research Center. I'm co-director of this center. And we're devoted to research excellence to cure muscle diseases. And we meet every month to form a research community for muscle diseases. And I'm applying our muscle stem cells in a wide range of different muscle diseases. And it's very exciting to be part of this growing group. So it really takes a team to translate skeletal muscle stem cell therapies for skeletal muscle wasting disease. And I'm very glad to have a lab here with PhD students. My lab is located here in Sue and Bill Gross Hall, which is a CERM Institute. And what I really like about the Gross Hall is that we actually see patients. Our first floor has the CERM Alpha Clinic, and a lot of times there's people down there that are undergoing different types of stem cell therapies. And it's really great to see that interaction for stem cells with patients. Yes, so just there's more information if you would like to get a hold of me on my lab website or Twitter or by email. And I thank you very much for your time and attention today.
Thank you so much, Dr. Hicks. That was excellent. Do you want to stop sharing for a second so we can see each other to ask questions? Wonderful. I actually have a question. It's a little bit naive, so forgive my naivete, but how much material do you need in terms of the stem cell in order to regenerate how much muscle?
Oh, that's a good question. So if you have a true stem cell, people have shown that even one stem cell can cure several fibers because that stem cell can proliferate and then differentiate into muscle. So the real trick there is to get the bona fide muscle stem cell that you only need a few. What we transplant, so the cells that we make from pluripotent cells, as I mentioned, they must go through each of the developmental steps. And so what we've actually discovered is that we make more of a fetal-like stem cell, which is not quite as powerful as the adult equivalent. We're still working on that. So currently what we do is we transplant 1 million muscle progenitor cells into a mouse tissue that's maybe 1 or 2 centimeters wide. And so we can get that to cure at a very high rate when we do high density injections like that. So for a human, I think a lower bar than say for Duchenne is to go after single muscle disorders. For example, rotator cuff tear injury, where you have 1 or 2 muscles that are chronically damaged, would be a more appropriate approach for the initial muscle stem cell therapies.
And is it something that you do this once and then the muscle forever knows how to correct itself, or do you have to keep doing it?
So that's a great question. And the goal is once. The goal is to do it one time. They tried in the 90s to do this where they took adult muscle stem cells from, say, a parent. They expanded those in a dish, and then they transplanted them into a kid with a muscle disease. And those didn't work. And the reason why is because the cells lost their stem cell expression when they expanded them indefinitely from an adult. And so when they transplanted, they worked for a couple of months. And then as the muscle turned over, they were pretty much not able to self-renew. So if we can get those PAX7 cells into the muscle to self-renew, the goal is that it's a one-time cell injection and it's there permanently.
Wow. Other questions? And please, everyone, if you have a chance, the survey has been added into the chat. If you could please complete the survey. And we have a question from Armando Vialta. I'll just read it. Is the altered migratory properties of the stem cell in dystrophic muscle attributed to a deficit in the stem cell itself, or an alerted environment that changes the behavior of the stem cell?
That's a good question. So we took wild type stem cells and so what we did is we took mice that were either a healthy normal mouse or a dystrophic mouse, the model for Duchenne muscular dystrophy, and we took the skeletal muscle and we decellularized it. So it was just the native extracellular environment. And in Duchenne, it's very fibrotic. You have these lesions that are fibrotic lesions. And when we took our stem cells and grew them on top of them to see what they would do, to see whether they could regenerate and fuse, what we found is that the wild type cells on top of the wild type matrix would be able to integrate with it. And they could remodel the matrix and they could break it down, and they would kind of make it their home. On the dystrophic environment, they could not break through it. They would just crawl along it like little bugs. You could see them crawling along on top of it, but they weren't able to integrate with it. And that had blocked their ability to differentiate and fuse to make muscle cells on top of that matrix. So the matrix of the dystrophic environment is actually, you could think of those fibrotic lesions as actually like a moat or like a wall that blocks the stem cells from getting in there to repair it. So we've talked a lot about how the muscle stem cells actually exhaust, but the environment plays a really important role for their ability to regenerate as well.
I have a quick question. Michael, thank you so much for that. I'm going to have to watch it over again because there was so much information in it. And I really appreciate both Armando and Dara's questions because they had their questions I was going to ask too. But I'm curious about, where do you get the stem cells in the first place? Are you taking them from an individual who needs treatment? And if that's the case and they replicate, wouldn't they just replicate part of the disease process and continue that disease? And of course, coming from the field of obstetrics, stem cells were a big deal. And so where are you getting the wild cells? Where are those wild cells? And then where do you start? Where do you actually get the stem cells in order to do all of this?
Great question. So for all the diseased stem cells, I actually got them from UCLA, my colleague. So the Center for Duchenne Muscular Dystrophy at UCLA actually has a clinic where boys with Duchenne, because it's X-linked, they come into the clinic and they get a skin biopsy. You can actually get stem cells from blood as well. So they get the skin biopsy, which gets your skin cells that are called fibroblasts, and then you can grow those in a dish, and then they reprogram those into iPSCs. And so there's a bank of them there. And so anyone can reach out to my colleagues at UCLA and get the Duchenne muscular dystrophy iPS-derived cells. We actually have something here too at UC Irvine through the Alzheimer's Disease Center. They have a bank of induced pluripotent stem cells that are from healthy individuals and also from Alzheimer's patients, but you can get the healthy ones if you prefer. And then you pretty much just sign this this material transfer agreement with the Alzheimer's disease, and they'll give you a vial. Then because the cells can expand indefinitely, you take the one vial and make as many as you need. But yeah, there's several different biorepositories that we can get. We can actually do it in our lab as well. You need like appropriate IRBs and things to do that, but it's much simpler to just get it from the biorepository.
Yeah, it's totally fascinating. And you're right. Everywhere in the community, you hear about stem cells, and there's people taking sea algae because it'll increase their stem cells. So I'm really glad you put that one slide in about it. There's no benefit. So I am going to look at this again because it was super interesting and lots of deep information. But thank you.
You're welcome.
Vince, last question.
Yeah. So Pam, I just want to come back to your question. Was your question about, we have a child who has Duchenne's muscular dystrophy? It was the question, where do we get the healthy cells to repair the damaged muscle? Was that really your question? Because Mike was really talking more about, cells that you could get to, I think, use for studying experimentally. But I think Pam's question was more directed at the ITS cells, Mike, and how you go about correcting these damaged cells that you've taken from an individual. Is that what your question was about?
So let me go back to that. There's different types of ways you can genetically alter a cell in a dish. And one of those, as I mentioned, was this new technology called CRISPR-Cas9. And so you can repair the mutation in that cell. And then the goal is to give that cell back to the person once the mutation is cured. And so that would actually be what they call autologous cell therapy. So you think of a liver transplant. You take a liver from a donor and give it to another person, and you're on immunosuppressions the rest of your life. This is not that. This is taking yourself, correcting yourself in a dish, and then giving yourself back to you, which would be autologous, and you would not need immunosuppression. It would be recognized as self. And so that's really the goal of our lab, and actually multiple labs here in the Stem Cell Center and elsewhere, where you can do this type of self therapy, personalized medicine.
Well, thank you. Thank you so much. One more round of applause. Thank you, Dr. Hicks. That was excellent. And now for our last presentation, Dr. Shiu is an assistant professor of dermatology, also in the School of Medicine here at UCI. And her talk today is identifying characteristics in stable vitiligo skin. And let's see. Perfect. We see the share, and thank you. Sorry.
Sorry, my screen froze.
I'm sure you're trying to do the share. Perfect. Oh, you're almost there, I think. Yes, excellent. Thank you.
Great. Hi, everyone. My name's Jessica. Thank you for that kind introduction. I am a dermatologist and scientist here at UCI, and it's a great honor to present some of my work looking at characteristics in stable vitiligo skin patients. So I just wanted to start with a basic introduction to myself and why I'm particularly interested in this skin condition. We know that our immune system is very powerful. On a daily basis, it keeps us safe from bacteria and viruses that we encounter. But we also know that sometimes the immune system can be dangerous and attack ourselves, leading to very common autoimmune issues like lupus or rheumatoid arthritis that we see in patients. In clinic, I often see patients with inflammatory skin conditions like psoriasis, vitiligo pictured here, and eczema. But I also see patients that are on the flip side that have a skin cancer like melanoma, basal cell skin cancer, where the immune system is failing to recognize the cancer to get rid of them. So I'm really interested in understanding why, our immune system can attack certain cells in certain conditions like vitiligo. And ultimately, I'm interested in understanding how we can improve treatments for both vitiligo patients, but also harnessing this understanding that our immune system is powerful to also treat, patients that have skin cancers. So just a basic introduction as to what vitiligo is. You may have already seen some of these patients, but maybe did not know what this condition exactly is. So it's actually one of the most common skin condition that we see in clinic that results in color changes of skin. And so very often in patients that have darker skin types, you can really see the contrast where they have white patches of skin. And around 1 in 100 people are affected. And most famously, Michael Jackson had this disease. And pictured here, I'm too old to know who she is, but I'm told she's very famous. She's a model, Winnie Harlow, that is now, really speaking out for vitiligo. We know that this condition can affect both genders and all ethnicities, but for our darker skin type patients, it has more significant effects, often, especially psychologically, because it really impacts their identity of self. And in certain cultures, like Indian cultures, it can even affect marriage proposals and society standings and job opportunities. And so vitiligo really is an autoimmune skin condition where your immune cells, a type of immune cell called your T cell, is attacking the pigment producing cells called melanocytes. And so clinically, it looks like this, where you can see literally black and white differences. And so a lot of times when our patients first develop this disease, it is a very active stage where your immune system is very active and there are many new spots developing over time and expanding quite rapidly. But over time, a lot of our patients then stabilize for unclear reasons where these white patches kind of stay white over years if they're not treated. And this is what we call stable vitiligo, where really we don't see a lot of immune cell changes anymore, but the skin stays white. We know clinically that it can be associated with other autoimmune conditions, especially thyroid disease and other like autoimmune diabetes or lupus as well. So to really understand, what vitiligo looks like, we need to understand what the skin looks like in different conditions. And so this is a picture indicating that our skin really is a community, that we may not think of that way, but this is how I think about skin. And so there are lots of different components that make up this community and neighborhood. And you can imagine, even though the skin is one organ, it has many different environments that encounters. So for example, the bottoms of your feet, just even running your hands over your feet, you can feel that the skin is different there than the skin under your armpit or your skin on your face. And so even though it is one organ, it behaves differently in different areas. And because of these challenges that the skin is faced with, it is always regenerating. And kind of as Dr. Hicks had mentioned, the skin also contains a lot of stem cells to maintain the barrier in the different communities that it lives in. And so we know that in skin that the bottom layer of cells is always growing and replenishing the top layers. And this is called the basal cells. We also know that there are other, cell types that are present in the skin called melanocytes, and these are the pigment-producing cells, and these are the cells that produce pigment, and then they actually transfer the pigment to the neighboring skin cells to create an even skin color that we see on our cells. At the same time, there are immune cells that enter in and out of our skin to keep us safe from bacteria and viruses and other environmental allergens that can enter, but they are overactive in vitiligo. And what happens is they come in and attack this pigment-producing cell so that no more pigment is produced and the skin becomes white. And so the current treatment strategies we have for vitiligo targets these different neighborhoods in the skin community. The first most common type of treatment is to target the immune response, whether it is an oral steroid or a topical steroid to suppress the immune cells that are present. And the second part of it is to aim to bring pigment to come back to this area. And often we do that by phototherapy, which induces signals in melanocytes, the pigment producing cells, to come back to the neighborhood and then start that transfer of pigment process again. There are other strategies that people are also trying currently for vitiligo where they use either antioxidants or when you have a lot of your skin being white, it's actually more convenient or cosmetically better for some of our patients to actually just bleach their whole skin, which is what Michael Jackson is believed to have done. And then in certain cases, we also, here at UCI, we're actually one of the main sites for doing this procedure on the West Coast, where we surgically transplant normal skin from the same patient to the diseased skin. And that process can actually also lead to some pigment reproduction. You may have heard this in the news, but there has been a lot of excitement in the dermatology and skin community because the first topical cream was recently, for vitiligo, was recently approved by the FDA. And this medication is called Cellular and mainly suppresses the immune system, but is not a topical steroid. So it doesn't carry some of the harms that we often think of with topical steroid, mainly skin thinning for our topical formulations. And so it's very, it's safer to use in the long term, especially on areas that already have thin skin, such as the face. But even in this instance, only 1/3 of our patients fully respond. And a lot of times, our patients are left with, partial, what we call partial repigmentation, where you have color coming back in some areas, but not all of it. So it's really uneven, and that, is still a problem. And so, as I alluded to, the problem really is that all our current treatment strategies together are not 100%. At best, it is around 50 to 60%. And a lot of times, it takes up to more than a year to see responses with whether it's phototherapy or topical treatment. And even when you see a response, as you can see on this patient's leg, it's uneven. We have areas where there's color coming back, and colors not coming back in certain areas. And we really don't quite understand why that is the case. And this is just another picture demonstrating this issue on one of our patients' face. And so when I first started this project, we were really just interested in looking at human patients and understanding, why in some of our patients, the skin stays white for a long time. And that was our starting point. And so, we know that, what are some of the factors that can determine, what determines skin color in our skin. We know that the immune system, for example, is present along all the skin. It's in fact actually one of the most abundant cell types that's present in the skin. But why is it that in the same leg, of a patient, there are some spots that stay white and in other spots, the color can come back? And we really think that understanding this, fundamental difference in the same area will help us understand vitiligo better and how to treat it better. So, one of the potential problem is that the immune system is more active in certain areas, such as this area where it stays white. And really, if so, what is it that's making it more active? Are there signals that are being produced that makes the immune system more active in this area? On the flip side, it could actually not be, it could not involve the immune system. It's possible that the immune system is actually quiescent, quiet and not really that active in these areas, but it really is mainly that the pigment producers are not coming back in certain areas. So this area stays white, whereas the pigment cells are coming back in this area. So the color is coming back there. But, we don't quite understand, what these signal differences are even within the same area of skin. And so, is there, the way I guess I think about it is it's kind of like the Nextdoor app where there's a lot of drama, but there's a lot of reports about coyotes and missing cats, and so, is there a signal that's saying, hey, there are coyotes here, please come over here, please stay away from here. Making the skin stay white and the lack of these signals so that these cells come back in these areas to bring the color back. And so one of the cell types that we really think is important in linking, the both possibilities, the immune cells and the melanocytes, are actually just our regular skin cells that are present on our skin. They are the most abundant in our skin, and they make up the basic skin architecture. And we already know from other people's research that they communicate with both cell types normally. And a lot of work has been done trying to look at its role in vitiligo, but most of them were done either with cells on a dish or with mice. And so there was some information that was missing in real patients. And so one of our goals was actually to map, these skin characteristics in our stable vitiligo patients using two different approaches. One is advanced imaging, and the second one is genomics. And so this is just a picture actually of the device and one of our researchers' arm here that you can see. And this is the imaging, the MPT Flex machine that we use to gather some of this data. And it really, is a very gentle process. It's a scan, it's almost like an ultrasound. We're letting technology do all the work here. There's no biopsy, there are no dyes, there's nothing. We just put a little mirror piece on your arm and then we image it, and we can generate images that on the computer to be analyzed. So when we analyze these images, and these are just some representative images, we can actually measure how our normal skin and vitiligo skin use energy differently. And so we know that normally skin cells at the basal layer, as part of the renewal process, they're using a lot of energy in a process called glycolysis. In our vitiligo skin, the cells, for whatever reason, actually do not switch to this energy utilization revitalization. And we think that this is an important process, mainly because in our patients that actually respond to treatment, they revert back to this normal skin process of using glycolysis, whereas in our patients that do not respond to treatment, they actually stay not using glycolysis. They do not switch back. And so, we had some hints from our non-invasive imaging of actually a lot of patients that, our normal skin cells are involved in this disease process. But we really didn't know what the molecular features were. And so we wanted to be able to identify molecules that were important for targeting treatment, for treatment targeting purposes, and also just to understand, what is exactly happening in our stable vitiligo patients. And so to do this, we decided to do single cell RNA sequencing, which is a genomics approach that allows us to look at different cell populations in skin. And so we had 6 vitiligo patients that were generous enough to provide skin samples from their normal skin as well as their vitiligo skin. And we isolate the skin using actually what we call a suction blister. So it's like a warm teacup being placed on your skin and negative pressure is applied to generate a little blister. And we take that blister, including the blister fluid, which contains the immune cells, and then look at it with single cell to identify unique cell populations that might be present and can be targeted for treatment. So when we did that, we found, and this is just a diagram showing the different cell populations that we see in normal skin here on the left and vitiligo skin here on the right. Each dot roughly represents a cell, that we see in that area of skin. And so as expected, we see kind of these different populations of skin cells in our normal skin. And then we also see some immune cells present and melanocytes. But in vitiligo skin, we actually see this enrichment of these unique skin cells that we called vitiligo keratinocytes that are not present in normal skin. And so we were really interested in why these cells were there and what they were making that could perpetuate vitiligo. Sorry, it's frozen. So when we look at what signals are being released from these vitiligo skin cells, we found that they were releasing more inflammatory signals to immune cells. I'm sorry, I don't know why that diagram didn't show up. But basically they were releasing these molecules to recruit more immune cells to the area. And then on the flip side, they were also not releasing the signals that we normally see that are important to recruit the pigment reproduction. And so, when we look at this, when we look at this in our vitiligo patients, what we have done is we've combined advanced microscopy and genomics to look at patient skin characteristics. We know that in normal unaffected skin, normally there is a good balance between the skin cells, which are the keratinocytes, with the immune cells, mainly the T cells here. And also there is a good communication between the keratinocytes and the pigment producing cells so that we have even skin tone in normal skin. What happens in our stable vitiligo patients is that for whatever reason, this unique cell population of keratinocytes arise and they are overactive in communicating with the immune cells. And then on the flip side, they're not communicating with melanocytes. So you can imagine this is just the same satellite image of New York City, where in normal skin, you get normal neighborhoods, but here there's a lot of alert signal going on to the immune cell, and then also a lack of signal to the pigment producers so that the skin stays white. And so the question really arises whether we can target these unique skin cell populations for treatment down the road. But to really demonstrate that, we're now focusing on, if these vitiligo skin cells are actually important in determining the repigmentation process. So we're focusing on in the same area of skin of our patients, isolating skin cells from the treatment responsive areas like this one, where you can see pigment coming back, versus treatment unresponsive areas that stay white, and using similar processes to look at, what determines why color comes back in some areas and not the others. And with that, I will end by thanking all the people that has helped to support my work, mainly in Anand Ganesan's laboratory, all the members in the lab, and also our close collaborator, Mihaela Balut, that helped us set up a lot of the imaging studies, and UCI Department of Dermatology, who has supported, who has provided, referred patients to us for part of the study, and also the UCI Skin Biology Resource Center that provided a lot of funding along with NIAMS.
Thank you so much for such an excellent presentation. Really excellent. Thank you. I have a question. Questions? Sure. I have a question. Thank you. So clearly for many people, vitiligo is visually, unpleasant. For some, it may not be. But is there underlying disease or illness that also one needs to worry about, or are you predominantly solving a I don't know how to say it, but can you talk a little bit about that piece?
Yeah, so, vitiligo, if you're asking if there's any internal implications in terms of like other organs being affected, we do see, a lot of times the thyroid being affected in our vitiligo patients. And so that usually, if we see vitiligo patients, we will check their thyroid hormone levels and things like that. And that occurs in around 10% of our vitiligo patients. Most of them are, only have skin limited disease. And so, but, it's actually has a lot of, people have a lot tied to their skin color. There's a lot of psychological impact there. So a lot of our vitiligo patients have depression or anxiety because of their skin condition, and it affects how they interact with their friends and family. And a lot of, traditionally, a lot of dermatologists have kind of dismissed this process as just a cosmetic condition, but really it's a lot more than that. It's not just cosmetic. And now with the advent of immunotherapy for melanoma, we're actually seeing more patients develop vitiligo because of their cancer treatment. And it actually is a good sign in a way, because it's a positive prognostic sign. Our melanoma patients on immunotherapy that have vitiligo actually do a little better than our patients that don't develop vitiligo. And we're certainly also interested in using some of the techniques that we've used to understand that process in our melanoma patients. But this is just kind of the first piece of that work.
Thank you. Dr. Villalta?
Hi, Jessica.
Hi.
Great talk.
Thanks.
I had a question, which I hope that there was a point where I was transitioning from home to the office, so I hope I didn't miss this. But the question is, I appreciated from your work that vitiligo is an autoimmune disease. And then folks are taking the approach of either a skin transplant, I think is what you heard, or even stem cells to try to restore the melanocytes. I would imagine that those things would be subjected to a similar autoimmune attack. So is it fair to say that, these type of replacement therapies would also benefit, or you need a type of immune tolerizing therapy that's used in conjunction? And if that's the thinking, what would you propose in terms of immune tolerizing therapy?
Great question. So actually, one of the reasons why we were interested in stable vitiligo is that we know in the initial acute phase of the disease, there's a lot of T cells that are present. So when you take a biopsy, you can actually see the T cells. But in stable vitiligo, where you see just these milky white patches of skin that have been there for years, there's actually just 1 or 2 T cells there. And so those are the patients that we select for the transplant process, where we take actually normal skin from the same patient, from usually the thigh or an area that's hidden that you don't see. And we take skin from that area and put it into the disease skin. And in that case, it actually does fairly well. Most of our patients at least partially respond. Of course, there are some that don't respond. And we do think in the patients that don't respond, it's probably because the immune system is attacking it. But, we have to show that.
What's interesting about that observation that you're sharing is that the white skin presumably no longer has the antigen that's driving your T cells, right? And yes. I wonder, it's hard to probably, maybe this has been checked, but the tissue or skin that's being transplanted that has color, do we know if any of that is devoid of any autoreactive T cells? Like what if you're actually putting a piece of skin that has the autoreactive T cells?
Yeah. And so, I think that's the other possibility is when you put in the transplant, right? It could be that the disease area is attacking it. And then the other piece is that, there are more T cells there to begin with, and it's not working. It certainly is a possibility. But one of the things that's really interesting is that, yes, the area that is totally white is devoid of T cells. But when people have looked at the edge, and this is true for a lot of vitiligo lesions, but if you look at just the edge where in our active patients, that's where the T cells are, and you can actually still isolate autoreactive T cells. But you're right in that there's no pigment being produced anymore to stimulate as an antigen, But one of the things we think actually is happening is that the keratinocytes, the regular skin cells, probably has some form of innate memory type of thing, where it alters that environment, so that it either there are signals that are inhibiting more immune cell infiltration in that area. So we're actually doing some spatial transcriptomics to look at those regional differences to really hone in on what the local signals are to understand what determines, why color comes back in some areas and not others.
Pam?
Yeah, so thank you for this talk. It was just super interesting. But I'm wondering, because you're working with the stable patients, is there work being done when the vitiligo is just starting to stop that immune response, nip it in the bud, so to speak?
Yes. So that, John Harris at UMass does a lot of the active vitiligo work. And so, they are the ones that have shown, the role of T cells, and they're really trying to control the T cells, and I think that certainly is a very important part of the disease. When I see really active patients in my vitiligo clinic, we start them on pulse dose steroids to stop the process, and actually works fairly well. But what happens then is they are still left with these white patches, and we still have to fix that problem, the white patches of skin problem. And that's where a lot of this work has a lot more relevance.
Jessica, I forgot to ask one quick question. Do we know what the autoantigens are for the T cells?
Yeah, so it's MART1, a lot of the melanocyte antigens. MART1 is one of the main ones, and there are some other melanocyte antigens in there as well. And that's kind of why people think that vitiligo, melanoma patients that develop a vitiligo do better, because they're targeting part of the antigens that are also present in melanoma.
Okay, thank you.
As we wrap up these three presentations, if we can give them all one more big round of applause virtually. And these were just such excellent presentations. And I think really highlight the cutting edge innovation that is occurring here at UCI. It's the best, the three of you represent the best of the research that we do here. And we're very grateful and for you to come and present to our community campus research exchange. So thank you for taking the time and explaining your work to us. This was just incredible. If it's okay with the three of you, we'd like to post your talks on our webpage. If you have any concern, please just send it in the chat. You don't have to say publicly now, but we will also be sharing with all three of you the results of your survey and also the recording. So you will have that for you. And thank you again. And thank you, Vince, really for your support for doing this. I know how much work went into supporting these 3 talks. So it was just really fantastic for our group. Thank you. And thank you to everyone, and have a wonderful weekend.
Thank you so much.
Thanks, Sarah. Thanks, Robin.
Thank you.
Thank you.